stepped acid hydrolysis of starch. The theoretical framework. In this class, we learned more about how it works and what method is used for starch hydrolysis. One method of modification is acid hydrolysis, where starch molecules polymerize or break bonds more or less randomly. Initially producing large fragments which reduce their viscosity. Starch when heated with acid. C. Um, it breaks down with the information of fragments of different sizes called dextrins. And they differ from each other in terms of their molecular mass and the collaboration they take on when treated with Lugol's solutions. Um, acid base hydrolysis. In this reaction, water splits into a hydroxylase ion and a proton, which is immediately hydrated to form a hydronium ion. Thus, pure water manifests this reaction spontaneously. Strong acid strong base salt hydrolysis. When a salt from a strong acid and a strong base is diluted in water, almost no hydrolysis occurs, so the dissociation equilibrium of wet water is not disturbed. The pH in this case will be neutral. Strong base, weak acid salt hydrolysis. The anion from the salt, from the weak acid and strong base, and a proton from the water combine, releasing hydroxy ions, due to which the resulting pH will be basic. Weak acid, weak base salt hydrolysis. The cation on the salt from the make base combines with water to release a hydronium ion, and the anion on the salt from the weak acid combines with water to release a hydroxyl ion. The resulting pH will depend on the amounts of hydronium and hydroxyl ions produced. If more H30 plus ion is produced, O oxygen or OH ion, the pH will be acidic, and if more OH ion is produced than H30 plus ion, the pH will be basic. On the other hand, if the quantities of both ions produced are equal, the resulting pH will be neutral. Hydrolysis of amides esters. In these types of organic substances, hydrolysis can occur in an acidic or basic medium. In the case of esters, the hydrolysis in an acid 1 and basic 2 medium, generating carboxylic acids and alcohols. The process of hydrolysis of esters is also called saponification, hydrolysis or triglycerides or to obtain swaps. On the other hand, amides generally hydrolyze in an acid medium, decomposing into amines and carboxylic acids. Enzymatic hydrolysis, it is the hydrolysis that occurs in the presence of enzymes, organic compounds that generally increase the speed of chemical reaction, called hydrolysis. For example, urea amidohydrolase is an enzyme involved in the hydrolysis of urea. Polysaccharide hydrolysis. Polysaccharide sugars can be hydrolyzed and broken down, breaking their glycosidic bonds, which are bonds between monosaccharides to form polysaccharides into simpler polysaccharides, disaccharides, or monosaccharides. Disaccharides. In the hydrolysis process, hydrogen from the weather molecule bonds to the oxygen at the end of a sugar molecule, while the hydroxyl bonds to the end of the rest. The hydrolysis of polysaccharides is a process carried out regularly by life forms. Experimentation. For this, we use the following materials: 1% repeat. Experimentation. For this, we use the following materials: 1% starch solution, concentrated hydrochloric acid, 10 test tubes, Lugol solution, 10%, sodium plus oxygen plus hydrogen solution, and free links reagent. And process. In test tube, we pour 5 milliliters of starch solution and add 1 milliliters of hydrochloric acid. This is mixed and brought to a boil. After 1 or 2 minutes after the start of boiling, 3 drops of the hydrolyzed are taken and 1 drop of Lugol's is added to a tube containing 2 milliliters of distilled water. These samples are taken every 2 minutes and we perform the Lugol's test on the tubes containing distilled water until the reaction is negative and yellow color. The starch hydrolyzed is neutralized with sodium hydroxide solution and fillings. Test is performed. The presence of a red precipitate indicates the presence of reducing sugars and products of acid hydrolysis of starch.